Hello everyone, my name is Dominique Schwend, and today I will be talking to you about the DNA binding and application of the Split T7 RNA polymerase. First, to understand the split of the protein, I will talk a little bit about the full-length T7 polymerase, which, as you may know, is an enzyme that catalyzes the transcription process from DNA to RNA, being 8 times faster than the E. coli RNA polymerase. It is a monomeric enzyme that recognizes the T7 promoter and does not require coenzymes or cofactors to function, being widely used in molecular biology. To achieve the split, a group of scientists made a transposon modification that generated a cut at the 601 position, leading to two separate subunits without losing the enzymatic activity when they interact with each other. Seen in the figure, the sigma fragments, also known as the C-terminal split, contain the specificity loop, which is in charge of the DNA interaction and recognition, represented in red in the model, whereas in white and blue is represented the core fragment, also known as the N-terminal split, which is not necessary for the DNA enzyme interaction. Nonetheless, the transcription process cannot be carried out without it. After splitting the enzyme, they designed different sigma fragments that, with a few changes on the aminoacidic sequence, altered the region of the DNA that was recognized as the promoter, therefore obtaining orthogonal responses to each sigma fragment to specific promoter, as you can see by the fold induction. The two main reasons why I chose this protein are the fact that it was altered by a group of scientists that wanted to extend the use of it on molecular biology, seeing a potential on a well-known enzyme that had not been explored before. With that, they opened the doors for new applications of it to different techniques that have caught my interest. Being one of these techniques, phage-assisted continuous evolution, where you can direct the evolution of two proteins of interest, enhancing the interaction between them by the fusion of each one of it to a specific split of the polymerase that has been designed to be proximity dependent to show the enzymatic activity. When the affinity of the two proteins of interest is low, the probability of the splits interacting with each other will also be undermined, leading to low infective phages, since the protein that grants the infective capacity to the phage is encoded under the T7 promoter. Therefore, mutations that enhance this protein and protein interaction will be accumulated, since this leads to the active form of the polymerase and therefore obtaining high infective phages. And this can go on and on for different rounds of evolution, enhancing interaction between the two proteins of your interest, in this case represented as the antibody and the antigen. The splits can also be used on the design of biosensor platforms, which for time purposes I will not be talking about. Nonetheless, I hope you like this protein as much as I did, and thank you so much for watching. These are the references that I used, and I hope you have a great day.